Chapter 4. Early Phase Digestive Signaling. Early phase digestive signaling establishes the operating conditions for the entire system before the stomach begins its primary work. This phase extends beyond the mouth and into the initial interaction between the stomach and the upper small intestine, where neural and hormonal signals coordinate secretion, motility, and timing across multiple organs. The system uses this early window to align output with the nature and volume of incoming material. When this signaling is intact, digestion proceeds with coordinated precision. When it is weak or disordered, the sequence begins out of alignment and every downstream process operates under constraint. The stomach is not only a processing chamber, but also a signaling hub. As food enters, mechanoreceptors and chemoreceptors detect volume, composition, and physical properties. These signals are transmitted through neural pathways and local reflexes that adjust acid secretion, enzyme release, and the rate of gastric emptying. Protein content, fat presence, and overall load influence how strongly these responses are activated. When early phase signaling is functioning properly, the stomach modulates its output to match the demands of the meal, increasing acid for protein-rich inputs and adjusting emptying to ensure that material is adequately prepared before entering the small intestine. Hormonal signaling reinforces and extends this coordination. Cells in the stomach and upper intestine release messengers that regulate downstream activity based on what has been detected upstream. These signals influence pancreatic enzyme secretion, gallbladder contraction, and intestinal motility. They create a feedback loop that links the stomach to the small intestine, ensuring that each stage is informed about the state of the material it will receive. When this communication is precise, enzyme release and bile flow are timed to coincide with the arrival of partially processed material, allowing breakdown and absorption to proceed efficiently. The timing of gastric emptying is a critical component of this phase. The stomach does not release its contents at a fixed rate. It adjusts emptying based on how well the material has been processed and how prepared the small intestine is to receive it. When early signaling is intact, this timing is synchronized. The intestine receives material in controlled amounts that match its capacity for enzymatic breakdown and absorption. When signaling is disrupted, this synchronization breaks down. The stomach may release material too quickly, overwhelming the intestine with inadequately prepared substrates, or hold material too long, increasing retention and altering internal conditions. This phase also determines how effectively fats are handled. The presence of lipids triggers signals that coordinate bile release and pancreatic lipase activity. These signals originate early and depend on accurate detection of fat content. When signaling is strong, bile is delivered in a pattern that allows lipids to be emulsified into structures that can be absorbed. When signaling is weak or mistimed, bile release may be insufficient or delayed, leaving fats in forms that resist transport across the intestinal lining. This alters both nutrient absorption and the composition of material that continues through the gut. Disruption in early phase signaling often originates from deficiencies in the stages that precede it. Weak sensory activation, rapid eating, and suppressed parasympathetic tone reduce the strength of the initial signals that drive this phase. As a result, the stomach and intestine operate with incomplete information and reduced coordination. Each organ continues to function, but the system as a whole loses synchronization. The material that moves through the sequence is processed under conditions that vary from one stage to the next, increasing inefficiency and variability in outcomes. Restoring early phase signaling requires strengthening the inputs that initiate it and reestablishing the conditions that allow it to operate. When sensory engagement is sufficient, parasympathetic signaling is present, and mechanical preparation in the mouth is adequate, the stomach receives clear and sustained signals about incoming material. Hormonal and neural pathways then coordinate downstream responses with greater precision. The sequence begins with alignment, allowing each stage to prepare the next without relying on compensation. Saliva, enzymes, and anticipatory signaling. Saliva initiates digestion by combining early chemical breakdown with a signaling function that prepares the rest of the system for incoming material. As food enters the mouth, salivary glands increase output under parasympathetic control, delivering fluid, electrolytes, and enzymes that begin to act on specific components of the meal. This activity is modest in terms of total breakdown, but it establishes the first chemical interaction between food and the body. At the same time, the presence of saliva and the act of chewing amplify neural signaling that raises gastric and pancreatic output in advance of swallowing. The system is not waiting to react. It is scaling its activity based on what is being sensed. Salivary enzymes begin the reduction of certain macromolecules while food is still in the mouth. This early activity alters the physical structure of the bolus and creates gradients that continue to influence processing after swallowing. Hydration from saliva disperses particles and reduces friction, allowing gastric mixing to occur more uniformly. A well-hydrated bolus exposes more surface area to acid and enzymes once it reaches the stomach. When saliva is limited, food enters in a drier, less integrated form that resists uniform exposure to digestive fluids. Portions of the meal may be processed effectively, while others remain structurally intact, creating variability that persists downstream. The signaling role of saliva is tied to its method of release. Salivary secretion is driven by neural pathways that also regulate gastric acid production and pancreatic enzyme release. As salivation increases, it reflects an increase in parasympathetic activity that extends beyond the mouth. This coordinated activation raises acid output in the stomach and primes the pancreas to deliver enzymes in alignment with incoming material. The system uses salivation as both a functional and informational signal. It indicates that digestion is about to proceed and adjusts downstream output accordingly. Anticipatory signaling extends this coordination further. Sensory input from taste and smell is integrated with the act of chewing to generate a sustained neural signal that informs the system about the composition and volume of the meal. This information influences not only how much acid and enzyme will be produced, but also when they will be released. The timing established during this phase determines whether gastric and intestinal processes will align with the arrival of food. When signaling is strong, acid production rises before significant gastric loading occurs, and enzyme release is synchronized with the movement of partially processed material into the small intestine. When signaling is weak, these processes lag, and the sequence begins with misalignment. This early phase shapes the efficiency of every subsequent stage. Proper salivary function and anticipatory signaling reduce the burden on gastric processing by delivering a bolus that is both mechanically and chemically prepared. They also ensure that downstream systems are operating at a level that matches the demands of the meal. When this phase is reduced, the system must compensate later. The stomach attempts to increase acid after fact. Enzymes are released into an environment that has not been properly prepared, and bile flow may not align with fat intake. These compensations increase variability and reduce overall efficiency. Saliva, enzymes, and anticipatory signaling therefore establish the initial conditions for digestion. They determine both the structure of the material entering the stomach and the readiness of the system that will process it. When this phase is intact, the sequence begins with alignment between input and capacity. When it is weakened, the system starts with deficits that propagate through every stage, increasing the likelihood of incomplete transformation and downstream instability. Brain, gut coordination before swallowing. Coordination between the brain and the digestive tract is established before food leaves the mouth, and this coordination determines how effectively the system will handle what follows. The act of chewing and sensing food generates continuous neural input that is integrated centrally and transmitted to the stomach, pancreas, liver, and intestine. This input does not simply signal that food is present. It carries information about volume, texture, and composition, allowing the system to scale its output in advance. By the time swallowing occurs, multiple organs have already adjusted their activity to match what is about to arrive. The vagal pathway serves as the primary conduit for this coordination. Signals generated in response to sensory input increase parasympathetic tone, which raises gastric acid production, primes enzyme release, and prepares bile delivery. These changes occur while food is still in the mouth. The stomach begins increasing its secretory activity, the pancreas adjusts its readiness to release enzymes, and the gallbladder is signaled to prepare for contraction. The system is aligning its capacity with the anticipated load so that the first stage of processing can occur without delay. This coordination also influences how the stomach will manage incoming material. Neural input from the cephalic phase affects both secretion and motility, determining how strongly the stomach will contract and how quickly it will empty its contents. When signaling is sufficient, the stomach is prepared to both chemically and mechanically process food as it arrives. When signaling is weak, the stomach receives material without having increased its output, and it must attempt to compensate after the fact. This delay alters the initial transformation and affects the quality of material that moves forward. The pancreas and liver are integrated into this early coordination through the same signaling network. Enzyme secretion and bile release depend on timing that is established before swallowing. When the brain-gut connection is active, these organs are primed to respond in synchrony with the movement of food from the stomach into the small intestine. This synchronization ensures that macromolecules are reduced efficiently and that lipids are emulsified at the moment they require it. When coordination is reduced, these responses become mistimed and the system loses precision. The strength of this coordination depends on the continuity of sensory input. Sustained chewing and engagement maintain the signal, allowing output to rise progressively. Rapid intake interrupts this process, shortening the duration of signaling and reducing its intensity. The system receives a weaker preparatory signal and begins digestion from a lower baseline. Each downstream stage is then forced to operate with reduced input, increasing the likelihood of incomplete processing. Brain-gut coordination before swallowing establishes the timing and scale of the digestive sequence. It determines whether the system will begin with aligned output or with a deficit that must be managed later. When this coordination is intact, the transition from preparation to processing occurs without interruption, and each organ contributes at the appropriate moment. When it is weakened, the sequence begins out of sync, and the effects propagate through every stage of digestion. How weak signaling propagates downstream. Weak early-phase signaling lowers the starting output of the entire digestive system, and that deficit carries forward through each stage as a compounding constraint. When sensory input and parasympathetic activation are insufficient, gastric secretion begins below the level required to alter incoming material effectively. Acid production rises slowly and often remains suboptimal relative to the volume and composition of the meal. Proteins enter an environment that does not fully unfold their structure, leaving peptide bonds less accessible to enzymatic cleavage. The stomach still mixes and churns, but the chemical conditions required for uniform transformation are not established early enough. This initial deficit changes the form of material that exits the stomach. Instead of a consistently processed mixture, the small intestine receives substrates with uneven levels of preparation. Some fractions are partially broken down, while others retain structural resistance. Pancreatic enzymes encounter this variability and must operate on substrates that are not uniformly accessible. Enzyme activity depends on contact with specific sites, and when those sites are hidden within intact structures, breakdown slows. The intestine compensates by extending exposure time or increasing secretory output where possible, but this compensation is inconsistent and often incomplete. Bile coordination is affected in parallel. Early signaling contributes to the timing of bile release, aligning it with the arrival of lipids that require emulsification. When signaling is weak, this timing shifts. Bile may be released in insufficient quantities or at points that do not match the presence of dietary fat. Lipids remain in larger aggregates that resist dispersion, reducing the efficiency of lipase activity and limiting the formation of transportable structures. As these partially processed fats move through the intestine, they alter the composition of the luminal environment and influence how other nutrients are handled. Motility patterns adjust in response to this altered substrate. The intestine uses feedback from the material it receives to regulate transit speed. When substrates are inadequately processed, the system may slow movement to increase exposure time, or in some cases accelerate transit if irritation or osmotic effects are present. These adjustments are reactive rather than coordinated. The sequence loses its predictable timing, and the relationship between secretion, breakdown, and absorption becomes less stable. Variability increases from one meal to the next because the starting conditions are inconsistent. Absorption efficiency declines as a direct consequence of these upstream changes. Nutrients that have not been reduced to their absorbable forms remain within the lumen, and the transport mechanisms of the intestinal lining are underutilized. Larger fragments and unprocessed components continue forward, increasing the load on distal regions of the gut. Microbial populations encounter substrates that would normally be absorbed earlier, shifting fermentation patterns and altering the chemical environment. This introduces additional variability and can amplify symptoms that are often attributed solely to microbial imbalance. The propagation of weak signaling is therefore not a single failure, but a sequence of dependent effects. A reduced signal at the beginning lowers acid output, which alters substrate structure, which affects enzyme efficiency, which disrupts bowel coordination, which changes motility, which reduces absorption. Each step inherits the conditions created by the previous one. Strengthening the initial signal raises the starting output and changes the trajectory of the entire sequence, allowing each stage to operate on material that has been properly prepared and aligned with the system's capacity.